Hi guys, this is Jason Matthew here again, and today we're going to be looking at lipids, and this will be part one, so there will be other parts coming up. So what are we going to be doing in today's class? Well, for one thing, we're going to be looking at the structures of some common lipids. We'll also discuss the functions of lipids. We'll look at what is a saturated fat and what is an unsaturated fat. We'll look to see how we name fatty acids, trends and physical properties of fatty acids, what are essential fatty acids, what makes a fatty acid an omega-3 or an omega-6 fatty acid, look at trans fat, triacylglycerol formation, and lastly we look at the hydrolysis of fats. So this is what we're going to be looking at in today's lecture. So as you can see here, we have some structures or some common classes of lipids. So we have fatty acids, we have the tag, triacylglycerol, phospholipids, steroids, glycolipids. And we're going to be looking at each one in more detail in this part as well as the subsequent parts to come. So fats and oils. Fats are generally solids at room temperature, while oils are usually liquids. So fats are generally solids, oils generally liquids. And we'll see later on that has to do with the type of hydrocarbon chains that these things contain, saturated versus unsaturated. So fats contain saturated hydrocarbon chains, while oils contain unsaturated hydrocarbon chains with at least one carbon double bond. Frequently, there are several carbon double bonds, and they are known as polyunsaturated. So just remember, we're going to look at it a little bit more later on, but a saturated fat has no double bonds. An unsaturated fat have at least one carbon double bond. Most of the times they'll have more than one carbon double bond, and we call that polyunsaturated. There's also waxes, and that's when a fatty acid combines with a long chain alcohol. It has some important uses in fruits. It's a natural protective layer in fruits, vegetables, and in some cases it's added for the appearance and protection. So beeswax, I think everybody's familiar with that, and that's just the chemical formula for it. So if you watch, you see here, this part of the molecule here would be the fatty acid component, and this would have been your long chain alcohol. Alright, so if we were to break this up, this part here would have been your fatty acid component, while this part here would have been your alcohol, your long chain alcohol to be more specific. So chain length. Short chain fatty acids are less than 6 carbons. Medium chain fatty acids are 6 to 12 carbons, and long chain fatty acids are more than 14 carbons. Most naturally occurring fatty acids contain even numbers of carbons in their chains. Functions and properties of fatty acids. They are a very good source of energy. They act as an energy reserve. Alright, any excess energy from carbohydrate, protein, and lipids are stored as triacylglycerols and adipose tissues. They provide insulation to the body from cold, and this is particularly important in hibernating animals such as bears and so on. It provides mechanical insulation, protects vital organs such as the kidneys and the liver and so on. So triacylglycerols provide insulation and storage for energy. hibernating animals generate a lot of triacylglycerols under their skin. Right? They store carbon as triacylglycerols instead of glycogen and starch. And there's a reason for that. Right? A more reduced form of carbon, right, will get more energy, about twice from oxidation. And the hydrophobic character does not necessitate carrying water weight. Because we know with carbohydrates, they had to have a lot of water with them as well. All right. Now, just to go back to this point here. All right. What we are saying is that fat is a more reduced form of carbon than, let's say, glycogen and starch. Because if you look at glycogen and starch, all right, or you just look at the monomer, which is glucose, you see that there's a lot of OH groups there. In other words, there's a lot of oxygen. But when you look at a fatty acid, with the exception of that carboxyl end, the rest of the fatty acid is just made up of carbons and hydrogens. So it means that a fatty acid is more reduced than, let's say, a carbohydrate. And when you look at when you have to burn something, basically, this is what respiration is. You're burning something, you're adding oxygen to this, right? You're going to get more energy if there's more oxidation. And because fat is in a more reduced form, then the amount of energy you're going to get from this combustion is going to be more than carbohydrates. So per gram, fat will give you more energy per gram than carbohydrate because fat is in a more reduced form than carbohydrate. Right? Fat also supplies essential fatty acid, and this would be the linoleic acid and linolenic acid. It helps in the formation of cell membranes, phospholipids, and also fat, specifically cholesterol, is the provides the steroid nucleus for steroid hormones. And we'll talk a little bit more about that in part 2. Alright, fat will also carry fat-soluble vitamins. They add flavor and taste to food. Right? If you are a fan of the Food Network channel, for instance, you will hear the chefs always making the statement that everything tastes better with bacon, and the reason for that is because bacon has a high fat content. So fat adds to flavor and taste. And lastly, it also gives a high satiety value. All right, meaning that you feel full when you eat a high fat content, because fats are digested slower. Right, so saturated fats, all the carbons are bonded to hydrogen. All right, so and for saturated fats, there are no double bonds present. All right, so if you remember your chemistry, you know that carbon is a tetravalent atom, meaning that it will have a maximum of 4 single bonds. And what we are saying that is in saturated fat, each carbon has 4 single bonds. There are no double bonds present. And saturated fats are long straight chains. They come from, most animal fats are saturated fats, and they will be solid at room temperature. Unfortunately, saturated fats also has a bad rep in that it contributes to cardiovascular disease, atherosclerosis, giving plaque deposits. On the other hand, we have unsaturated fats. And with unsaturated fats, there's a double bond being present. And most plant and fish fats, as well as vegetable oils, are unsaturated fats. And as we saw in the slide before, when you have the presence of double bonds, the phase that they will be in a liquid state at room temperature. All right, and this is the reason for that is this. Unsaturated fats have double bonds present, and to be more specific, they have cis double bonds. And what a cis double bond does is that it forms a kink right? So these double bonds, they form kinks, and they prevent the fat molecules from packing tightly together. And because of that, because they can't pack tightly, they are in a liquid state. So unsaturated fats, because they have that cis double bond, they are not able to pack tightly. Now with a saturated fat, as we saw before, they have no double bonds present. And because they have no double bonds present, there are no kinks in their molecule. And therefore, they are able to pack on as layers. And that's why they form the plaques that's in the arteries and they lead to heart disease and so on. So just remember, the bottom line is this. Saturated fats have no double bonds present. Unsaturated fats will have at least one double bond present. And that double bond will cause a kink in the structure. Remember, it's a cis. For naturally occurring fats, it will be a cis double bond. So here are some common fatty acids. So on the left-hand side, we have, so that is this here, we have saturated fatty acids. So we have saturated fatty acids here. And on the right-hand side, we have unsaturated fatty acids. So all of these fatty acids on this side will have at least one double bond. So you see we have divided them into monounsaturated, meaning that these guys here will only have one double bond. And then polyunsaturated, meaning that they will have more than one double bond. So some important ones to point out. Palmitic acid will be a 16 carbon saturated fatty acid. Stearic acid will be a 18 carbon saturated fatty acid. So it means that both palmitic and stearic acid, they'll be fatty acids, but they'll be saturated, meaning that they have no double bonds present. For monounsaturated fats, there's oleic acid, which is 18 carbon, and it has one double bond. And I just want to point out for now, linoleic acid and linolenic acid. These are polyunsaturated fats, and we will see that they are very important. They are what we call essential fatty acids. We're going to come to that very soon. And you see this sign here, which we're going to come to again, is the omega sign. So we're saying that linoleic acid is an omega-6 fatty acid, and linolenic acid is an omega-3 fatty acid. And we're going to see what all this means. This triangle here is delta. And what they're doing here, this is the delta designation. Alright, so we have linoleic acid, which is at 18 carbon. It has more than one double bond. Alright. Arachidonic acid, 20 carbons. Now, just a little side note here. Oleic acid. Alright, oleic acid is emitted by the decaying corpses of a number of insects, which includes bees and ants. And basically, what happens is that when this oleic acid is secreted from these dead bodies, it alerts other worker bees, if it's bees in that case, to remove the dead bodies away from the site. Alright? And also what it does is that the oleic acid, when it's secreted, it tells the other insects that to avoid this area, because it could possibly be an area of disease or where predators are. So insects will avoid that area where the oleic acid being secreted. So as a use now for us is that in some bug repellents, they use oleic acid. Oleic acid is present in a lot of bug repellents out there. So you can do some further research if you're interested in that. But I just thought I'd throw in that as a side note. So, although we're looking at these things and we've seen all these crazy formulas, there's a practical aspect, and this is what we learn from when we learn lipids. Alright, so remember, biochemistry, very interesting topic. And I hope you all are seeing that as we go ahead with all these lectures. Alright, so structure and nomenclature of fatty acids. And nomenclature just means you're going to be naming these things. How do we name this? Now, one of the things I want to point out here is that this is a typical fatty acid. Alright, so we have a typical fatty acid. Now, in a fatty acid, they're going to have two ends. And one end will be your carboxyl end. So you see C double bond OO. And but it's C double bond OOH, or if on a physiological pH, it'll be ionized, so it'll be C double bond OCO minus. Alright? So this is the carboxyl end. And then you have a long hydrocarbon chain attached to that carboxyl end. Right? So you have a long hydrocarbon chain, and a hydrocarbon chain just means that it's just made up of carbon and hydrogen atoms only. So on this end you have a carboxyl end, and on this end here, now this one here, this will be your methyl end. Alright? So let's just write that in. Alright, so here that will be your methyl end. So you see the CH3 present. And on the other end of the fatty acid, on the other end of the fatty acid here, this will be your carboxyl end. Alright, so you have two ends on the fatty acid, a methyl end, CH3, and a carboxyl end. And you see, this is important when we start naming the fatty acids, because according to what type of nomenclature you decide to use, you're going to either go from the carboxyl end or you go from the methyl end. So that's important. Alright, so here we have a fatty acid. So you see, alright, the one above, we showed all the individual bonds. You see all the bonds between the carbon and the hydrogen and so on. Or you could write it this way. Alright, so you just put the CH2, CH2, CH2 until you come to the, as I said, the methyl end, which is CH3. So you could write it as either one. Or you could even do a even more shorthand. And each CH2 now, every point here would represent a carbon atom bonded by two hydrogens. So each point here represents a CH2. Alright? So if you were to, so each point, alright, so this point here, oops, this point here, oh my, sorry about that. So this point here, all of these are basically CH2. So this here is where the carbon is. Alright, so you have a carbon here, and you have a hydrogen bonded to it, and the next hydrogen bonded to this one. So that's what that molecule represents. And we could even write it like this now. So all these here is the same stearic acid that we are drawing at. So this is the same molecule as this, which is the same molecule as this. And we could also write it as CH3. So that CH3 will be this N here. And then you have CH2. So that's CH2 here. And if you want, the CH2 is repeated time and over repeated 16 times. So this CH2, if you count each CH2 here, it will be 16 of them. So it's CH3, which is this fella here, CH2, which is 1, 2, 3, 4, 5, 6, 7, 8, and you continue, will be 16, and then you have the COOH, which is this guy here. So we call it stearic acid, or the systematic name will be octadecanoic acid. Now the shorthand now is that if this 18 here will represent the number of carbon atoms. Alright? So 1, 2, 3. If you keep counting, you're going to get 18 carbons. So this number 18 represents how many carbon atoms are present in the fatty acid. This 0 now represents the number of double bonds present in the fatty acid. But it actually must be the number of carbon-carbon double bonds. And if you look at this, or this, or this, you're not seeing any double bonds being present. So that's why you have 0. Alright? So let's try to give these two guys their delta designation. Alright? Now, if you think you have a hang of it, well then you pause the presentation right now and you write down what the delta designation is for this. Alright, remember you're always going from the carboxyl end. So try to see if you could do it. And then when you think you have it down and you've written it down, then press play and see if you got it right. So I'll be waiting right here patiently. Okay, so you think you have the answer. So let's see. Right, so for this one is 8. The delta designation is 8, 1, delta 3. That's the delta designation for this. So let's see how we got that number and be precise. Now with the delta designation, you'll be counting from the carboxyl end. So remember, over here, this is the carboxyl end. So the carbon on the carboxyl end will be carbon 1, and you keep numbering as you go down. So it's 1, 2, 3, and as you see, there were 8 carbon atoms. Alright, so there are 8 carbon atoms. So this 8 represents the number of carbon atoms present in the fatty acid. So in the one above here, we have, and it's numbered, 8 carbon atoms. Now you put the colon dot, and then there's a 1. And there's a reason there's a 1 here, because when you look through the molecule, there's this 1 carbon-carbon double bond. So there's a 1 carbon-carbon double bond. So it's 8 carbon atoms, and there's 1 carbon-carbon double bond present in the fatty acid. And the last thing you do, you put in brackets, you put delta 3. Now what do you think that 3 represents? The delta 3 represents the position of the double bond. So as again, this is where the numbering comes important. For the delta designation, you'll be counting from the carboxyl group. So you're going to give the carbon in the carboxyl group the number 1. So if you watch, we said 1, 2, 3. So we take the first small number 3 where the double bond is between. So we say delta 3. So this is international standard way of naming your fatty acid. So it means that if I give you this, you should be able to draw this above here, all right? Because you would know from this nomenclature here, this delta designation, that 8 means that there are 8 carbon atoms. There will be 1 double bond, and the double bond will be between carbons 3 and carbons 4. But we don't put the 4, we just put the first number, the smallest number of between the 2 carbons where the double bond is. So the double bond, if you look at the structure, is between carbon 3 and carbon 4. So again, to recap, 8 carbon atoms, 1 double bond, and it's between carbons 3 and carbon 4. So if you didn't get this one correct initially, that's fine. We're now learning how to do this. So what I'll do is to challenge you and then see if you could get this one correct now. So you should pause the video now and you write down an answer that you think is correct, and then check with me and let's see if you got it right. I'm sure you got the idea by now, but let's just make sure. So pause the video and you write down what you think the answer is, or what is the delta designation for this fatty acid. See what you come up with. Alright, so you should have gotten this answer, and let's see why. Again, you count the number of carbon atoms and you'll see there'll be 8 of them. You put the colon, and then you look for the number of double bonds. So when you look at this molecule here, you have 1 double bond, and then you have a next one. So you put 2 because there are 2 double bonds present. Then you do the delta sign, and now you're going to say, well, where are these double bonds found? And again, remember, when you're dealing with delta designation, you'll be numbering from the carboxyl end. So the carboxyl carbon will have 1, and then you keep numbering as you go along. So you see that the first double bond is between carbons 3 and 4, so we give 3. And in the other double bond is between carbons 6 and 7, so we give 6. We always take a lower number. Now, patterns in commonly occurring natural fatty acids. Naturally occurring fatty acids will have an even number of carbon atoms. And We will see that more in more detail when you do the level 2 courses, where we look at metabolism. All right, so you'll see that it's the result of using acetate, which is a 2-carbon molecule, as the building block. The location of double bonds in most monounsaturated fatty acids, the double bonds will be between carbons 9 and carbons 10. In polyunsaturated fatty acids, additional double bonds are at C12 and C15. As usual, they will always have some exceptions to the rules. And for naturally occurring fatty acids, as I said before, the double bonds are in the cis form. So characteristics of fatty acids. So I want you to look at this table. And as a good scientist, look at the table, look at what information is there, and try and see if you can't make a link with what you are looking at here. All right, so look at the table, look at the data that they're giving you. As a good scientist, try to make some kind of correlations, try to make some links between the data and see if you can't find any trends. All right, so pause the video and look at the table properly. And without me giving you the answer, try to find any trends that are present there. All right, so let's see. There are obviously a lot of information being given here, and you can see some clear trends. First of all, we are seeing that this here, C4, C6, C8, these are the lengths of the fatty acids. So this one has 4 carbons, 6 carbons, 8 carbons, 10 carbons. So in other words, we are increasing the length of the hydrocarbon chain as you go down. There was 4 carbons this fatty acid was made up of, and as you look at the bottom, it's 18 carbons. So we see as the number of carbons increase, we see that MP stands for melting point. That should have been degrees Celsius here. So let's see if we can write that here now. Alright, so right above here, that should have a little 0 there. So that's degrees Celsius, sorry. So the melting point, it goes from -8 to -4, 16, 31, 44, 54, 63. So in other words, as the number of carbons increase, we see that there's also a trend in the melting point. The melting point is increasing. All right, so that is one trend we've seen there. That as the number of carbon atoms increase, the melting point will increase. The other column gives us milligrams of fat per 100 mils in H2O. Now, I mean, there's some bad writing going on there that I must apologize for. First of all, especially if you're my student, please do not write mils in this very wrong way that they're doing it here in this table. This is from a textbook, sadly. All right, remember, mils is milliliters. So this L here should have been capital L. Well, obviously here's another typo, so it's H2O. Let's just correct that. Now, milligrams of solute in 100 mils of water is one way in which we measure solubility. So let's look at what's happening here. So in this column here, the melting point is increasing. But when we look at the solubility, we see 970, 75, 6.55, and so on. So you can see the trend, the general trend is that the solubility is decreasing. So let's summarize. All right, let's summarize this. As the number of carbons increase, all right, so as the number of carbons increase, the melting point also increases. But what happens to the solubility as the number of carbons increase? The solubility decreases. All right, so as the fatty acid, as the chain on the fatty acid grows, the melting point will increase. All right, but the solubility will decrease. So here we have 2 fatty acids. We have stearic acid and we have linoleic acid. Now, if you watch the two of them, all right, and you count the number of carbons there, you should see two of them both having how many carbons? See, count it up and see for yourself. I'm not going to give it away, right? So count the number of carbons present there. All right? Right, so yourself, if you can remember from the other slide, if you look at these structures here, you should be able to count up the number of carbons. I should rightly say that they have 18 carbons. So they have 1 plus 16, because this is 1 carbon taken 16 times. So it's 1 plus 16 plus 1, so that's 18 carbons. And if you do it here, it'll be 1 plus this carbon taken 7 times, so it's 1 plus 7 plus 1 plus 1 plus 1 plus 1 plus 1 plus 4 plus 1. And when you add up that, you should also get 18 carbons. So these two fatty acids have the same number of carbons, which is 18. But yet, when we look at the melting point, we see that this one, stearic acid, is significantly higher. It's 69.6 degrees Celsius, while the melting point for linoleic acid is -5. I suspect it's 5 degrees Celsius. So there's a significant difference in melting points. So they have the same number of carbon atoms. So then it has to be another factor. Now, what is the difference between these two when you look at the two structures here? So look at the structure. What you know about stearic acid and what you know about linoleic acid. All right. And when you look at these two structures, hopefully you saw that stearic acid is a saturated fatty acid, and linoleic acid is unsaturated fatty acid, meaning that there is the presence of double bonds. So stearic acid has no double bonds, while linoleic acid has 2 double bonds. So it means that if everything else is remaining the same, they have the same number of carbon atoms. The only difference is that linoleic acid has 2 double bonds. It means that the presence of double bonds lowers the melting point. So that's another physical trend of these fatty acids, that the more double bonds you have, the lower the melting point. So that's interesting. So you see from this graph, it confirms what we saw before between stearic acid and linoleic acid. All right, you see that, remember from your delta designation, this number represents the number of carbon atoms, and this number here represents the number of double bonds. So for this first fatty acid is 16 carbon atoms, 0 double bonds. This one is 16 carbon atoms, 1 double bond. And you see that just the presence of 1 double bond, it drops the melting point significantly. And you'll see the same trend as you go through the others as well. So basically, the melting point decreases as the number of double bonds increase. So that's some good stuff there. Right, so this is a very nice diagram here. If you watch, you see that we're looking at the trend with melting points of fatty acids. So on the x-axis, here, we have the number of carbon atoms. And on the y-axis, we have the melting temperature in degrees Celsius. Now, as you increase the number of carbon atoms, you see that the melting point increasing, and we saw that before. All right. Now, let's compare some 18 carbon fatty acids. So, again, this red line here shows that as you increase the number of carbon atoms, it's going to increase the melting point. And this is for saturated fatty acids. So this data was done on saturated fatty acids, meaning that none of these fatty acids that gave this data here contain a double bond. All of them was saturated. No double bonds were present. So let's look at the 18 carbon fatty acid now. So that will be stearic acid. So that's, as you see here, 18-0. There's no double bonds present. All right. Now look at what happens. But there's the presence of 1 double bond, the melting point drops. So that's oleic acid, where there's 1 double bond. So stearic acid, 18 carbons, 0 double bond. The melting point is roughly around 70 degrees Celsius. While with oleic acid, it could be around 15 degrees Celsius. So it drops. Now we go to linoleic acid. and that's now 2 double bonds present, and it drops, the melting point drops even more. And the reason for that is because it has more double bonds. So it goes to like minus 5 degrees Celsius. So my question is, if you let's say had, I don't know, 4 double bonds, where you think that if you had a fatty acid with 18 carbon atoms and 4 double bonds, where would it be? And you're right, it's going to go even lower. Alright? So like, well, linolenic acid 3 goes lower. And then if it had 4, it would be even lower. Alright? So basically, if you have the same number of carbon atoms, but you start increasing the number of double bonds present, then the melting point is going to decrease. And you should know why. Alright? It all has to do with what the double bonds do. What cis double bonds do? They prevent tight packing. They form kinks in the structure. And the more kinks you have, the less tightly packed they are. So they are easier to melt. So the physiological functions of lipids. The fatty acid composition of membrane lipids and adipose tissue triglycerides. Right? You're looking at the chain length and the degree of unsaturation. Because what organisms will do in their membranes, especially, that if they change the composition of the membrane, in other words, they change the chain length of fatty acid present, or they change the degree of unsaturation present, then they use that to regulate and maintain the fluidity of the membrane. Right, so essential fatty acids. And once you see the word essential in biochemistry and in nutrition, it means basically that the human body cannot synthesize it on its own. Alright? So most naturally occurring fats are a mixture of saturated, monounsaturated, and polyunsaturated fatty acids. Essential fatty acids are those that the body cannot synthesize on its own. They must be acquired from the foods we eat. Alright, so essential fatty acids, we cannot make them. So non-essential fatty acids, our body can make certain fatty acids, so they are not required in that. So these will be non-essential fatty acids. Alright, now what makes a fatty acid essential? Basically, our bodies cannot make carbon-carbon double bonds before the 9th carbon from the methyl end. So we must get these fatty acids from our diet. And life is easy. There are 2 essential fatty acids that we need to know. Alright, so our 2 essential fatty acids will be linoleic acid, which is our omega-6 fatty acid, and then linolenic acid, which is our omega-3 fatty acid. And remember, this is when we counting from the methyl end. So you're going to see more about that very soon. So essential fatty acids, omega-3 and 6. Alright? Omega indicates the position of the first double bond from the methyl group. And that is very important that we're using the methyl group. Because remember, let's recap a little. When we did the delta designation, that was from the carboxyl end. When we're doing the omega designation, you're using from the methyl end of the fatty acid. Alright, and you're looking for the first double bond. So the first point of unsaturation, meaning that the first presence of a double bond, if next to the 3rd carbon, it's an omega-3 fatty acid. If when you start counting from the methyl end and the first double bond you find is on carbon 6, so then it's an omega-6 fatty acid. And these omega fatty acids are very, very critical, right, in human nutrition and health, and health generally, right? So you will see that if you go to the drugstore, you'll see a lot of supplements that contain, and they will say that they have omega-3 and omega-6 fatty acids, and they give you all the health benefits of taking these supplements. So you can check that out. Right, so we're going to first look at linolenic acid. Alright, and this is an essential fatty acid. It's a cis isomer. Alright, and remember, as I said before, this symbol here is the symbol for omega. And remember, if you say it's an omega-3, if you say linolenic acid omega-3 fatty acid, it means that the first double bond from the methyl end will be on carbon 3. And if you look look at this. This is the fatty acid here. You have the carboxyl N, but that is not the N we're going to be counting because that's delta definition. Right now we're looking at omega designation. So it means that we're going to go on this side, which is the methyl N, and you count the number of carbons. So the methyl group will be designated carbon 1. So you have 1, 2, and then bam, 3. So the double bond is between carbons 3 and 4. So we say it's an omega 3 fatty acid. So linolenic acid is omega 3 fatty acid. So sources of omega-3 fatty acids: flaxseed, soybean oil, walnuts, all right, some dark leafy, dark green vegetables. All right, flaxseed, especially very rich in omega-3 fatty acids. All right, fatty fish, salmon, tuna, mackerel. All right. Then we have the omega-6, and the example here will be linoleic acid. Alright, and remember, as I said, this is omega. So if you count from the methyl N, 1, so this is carbon taken 3 times, so that's 1 plus 3 is 4, 5, and then bam, 6 is between carbon 6 and 7, the first double bond. Now, it doesn't mean that they won't have other double bonds, and more double bonds could be present, but the first double bond is at between carbon 6 and 7. So that's why we call it an omega-6 fatty acid. So sources of omega-6 fatty acids: seeds, nuts, common vegetable oils, also meat to some extent. Alright, how would the melting point? So this is a little recap now to see if you're still awake with me and if you understand what's going on. So how would the melting point of stearic acid compare to the melting points of oleic acid and linoleic acid? Assign the melting points of minus 17 degrees, 13 degrees Celsius and 19 degrees Celsius to the correct fatty acid and explain. Now, we already looked at it in the previous slide, so don't try to see if you could sort it out from your memory and what we did so far. See if you could do it on your own. Right? So you're going to be assigning these temperatures that they give you here to these fatty acids. And I have been kind enough to tell you how many carbons and how many double bonds are present. So you should be able to easily figure it out. So let's take a little time, write down what you think is the answer, and see what you come up with. Alright, so I'm pretty confident that you all figured it out. Stearic acid, 69 degrees Celsius. Oleic acid, 13, and linoleic acid, minus 17. It all has to do with the number of double bonds being present. Alright? Now, stearic acid is saturated, so there's no double bonds present. Linoleic acid has 2, while oleic acid only has 1. So the only 2 double bonds will obviously have the lower melting point. Now, if you didn't get that, what I would recommend is that you go back, rewind a little bit, and go back to the earlier section. You want to see it explained more in detail. Product of hydrogenation. Now, hydrogenation just simply means that we're going to be adding hydrogen across the double bonds. Alright? So basically, what we're looking at here is this. Now, remember what I told you before. The maximum carbon is a tetravalent atom, meaning that the maximum number of bonds, single bonds it could form is 4. So if we have, let's say, a carbon double bond. Alright? Each carbon here, and then it's going to be attached to something else, and it's going to be attached to something else. Alright? And it's going to have a hydrogen and a hydrogen. That's a carbon double bond there. So each carbon has 1, 2, 3, 4. 1, 2, 3, 4. Now, if I was to hydrogenate that means I will first break the double bond. So I'll get something like this. Alright, let's put back the original atoms there. Alright, so now you see all of a sudden, each carbon could have one more bond with something else. So let's just recap. Any carbon double bond here, each carbon has 4 bonds. 1, 2, 3, 4. 1, 2, 3, 4. But if we break this double bond here, so you just have 1 here, it means that this carbon has 1, 2, 3, but that means it could bind to something else, right? And this carbon could do the same thing, could now bind to something. So what we do is that we add hydrogen across the double bond. Now this is done a lot in the food industry, all right? And what it does is that you see, the thing with oils and stuff is that it is much more difficult to work with an oil, a liquid, than with a solid. So in the food industry, the solid is favored over the oil in terms of being able to manipulate it, as well as it increases the shelf life. All right. So the more hydrogen is added across, the more double bonds, the more solid it will become. Right, and it's a way in which we make margarine and so on. Now, how much double bonds are present can be answered if you use the iodine index. All right, so the iodine index shows the degree of unsaturation. All right, it measures that, and basically what you're doing is that something just like what you did with hydrogen. You're going to be breaking this double bond. So you're not going to have a single bond. So each carbon is going to accept one of the iodine atoms. Right? So what I want you to remember about the iodine index is that for each double bond, 1 mole of iodine is going to react with it. So let's remember that for me. So this is just the iodine index of some common fats and oils. And you will see that when you combine in the amount of double bonds here, so the more unsaturated the fat is, the higher the iodine index will be. Alright, so that's something to check out there. Now remember I was telling you before how we hydrogenate oils so that they add hydrogen, it's more to increase its shelf life and so on. There's no nutritional value for doing that. But here's the thing, by doing hydrogenation in the food industry, we form something called trans fatty acids or trans fats. And it's a little outside the amount of time that we have now. But what I would tell you all to do is to please go and read up about trans fats. Because if you don't know, there's a lot of media hype about it, but it's really nasty for us. So you should really read up about trans fats. It's very important to do that. So trans fatty acids are found in fried foods and in some margarines. Right? Trans fatty acids are generally considered undesirable since they increase the formation of LDL cholesterol and hence the risk of heart disease. And the reason for this is simple. Now remember as I was telling you all before, most naturally occurring, or I should say all naturally occurring fatty acids that have double bonds in them, it will be of the cis configuration. Alright, they'll be of the cis configuration. And probably I should mention what cis and trans isomers are. Now with cis isomerism, in a cis isomer, 2 of the same atoms are on the same side of the double bond. Alright? And so that's a cis isomer. And for a trans isomer is where you have the 2 same atoms on opposite sides of the double bond or across the double bonds like that. So so this is supposed to be a Y. Alright? So you see in this one the 2 hydrogens on the same sides as cis. In this one the hydrogen, the 2 similar atoms are on opposite sides or they're across the double bond. So we say that's trans isomer. Alright? And this is an unfortunate byproduct of when you hydrogenate fats. Alright, you turn some of the naturally occurring cis double bonds into trans double bonds. Now here's the thing. If you remember, and I'm sure you do, you will know that with cis double bonds, they form kinks in the molecule and they prevent packing, close packing. The thing is that with trans double bonds do not form any kinks. So the fatty acids are able to form layers upon each other. They're able to pack tightly And what that does is that it forms plaques in the arteries and so on, and it leads to heart disease. So it's very bad news for us. So, I mean, within the last few years, there's been a huge campaign to get rid of trans fat because they're just bad. Because they'll be like in your fried food, your fried chicken, they'll be in your potato chips and so on. And there has been a huge drive to get rid of trans fat in the food industry because it is extremely unhealthy for you. So fat metabolism, it provides more energy than carbohydrates, and we already spoke about that. And the reason for that is because fats per gram are more reduced than carbohydrates. So fats require a greater degree of oxidation to become carbon dioxide and water, that's respiration, than carbohydrates, because carbohydrates already have one oxygen for every carbon atom. And lastly, we come into the end. This is glycerol. All right. Now, if you look at this glycerol molecule, it has 3 hydroxyl groups. Now, what is a hydroxyl group? A hydroxyl group is simply an OH group. So, if you look, a glycerol molecule has 3 hydroxyl groups. 1, 2, 3. It also has 3 carbons. 1, 2, 3. So, a glycerol molecule is like this. It has 3 carbons. 1, 2, 3. And each carbon atom is attached to a hydroxyl group or an OH group. So OH, OH, OH. Now what could happen here is that, again, let's isolate this here, is glycerol, CH2, CH, CH2. And what happens is that a fatty acid, for every hydroxyl group present on glycerol, a fatty acid could combine with it. So if you watch here, there will be 1, 2, and then 3 fatty acids that could combine per glycerol molecule. So again, let's look at the stoichiometry here. For every 1 glycerol molecule, a maximum of 3 fatty acids could combine with 1 glycerol molecule. And the reason for that is that for every 1 glycerol molecule, there are 3 hydroxyl groups. Alright, so let's look here. So this is a straight chain form. So this is the glycerol molecule here. And again, it has 3 carbons. 1, 2, 3. And each one has a hydroxyl group. 1, 2, 3. And what that means is that for each hydroxyl group, it can react with a fatty acid. Now, which part of the fatty acid molecule will it react? Well, the functional group of a fatty acid is C double bond OH. In other words, the carboxyl group. So a carboxyl group will react with an alcohol group. Now, in this example here, we're using 3 palmitic acid molecules. But it could be any fatty acids. It doesn't have to be 3 of the same. It could have been different fatty acids. It could be all the same. 2 could be the same. Any combination of that sort. Once they are fatty acids and they have this carboxyl group, the carboxyl group is going to react with the hydroxyl group of glycerol. So again, these 3 could have been the same fatty acids or they could be different. The important thing here is that the fatty acid will have the carboxyl group, and that is what is going to be reacting with the hydroxyl group. But I guess just for simplicity's sake, we use the same fatty acid throughout. So you have palmitic acid. 3 moles of palmitic acid is going to react with 1 mole of glycerol. And that is the reason. The reason for that is because for 1 glycerol, there are 1, 2, 3 hydroxyl groups. Right. Now, when a fatty acid, reacts with a glycerol, basically what's happening there is that the carboxyl group of the fatty acid is reacting with the hydroxyl group of the glycerol molecule. And when a carboxyl group reacts with a hydroxyl group, the linkage that is formed is called an ester bond. Alright? Now, you're seeing that water is being released for each ester bond that is being formed. So, it's going to be 3 ester bonds going to be formed. For each ester bond being formed, 1 molecule of water is being released. So if it's 3 ester bonds or ester linkages being formed, then 3 molecules of water is going to be released. And you're taking 2 molecules, you're making a bigger molecule and a small molecule, so water is being released. That is called a condensation reaction. So just like when we build proteins or when we build carbohydrates, those were condensation reactions. This is also a condensation reaction. And if anyone ever asks you to draw the ester bond, remember to do something like this. It's going to be a carbon, all right? And then it's going to have a double bond O for the carboxyl group. Then it's going to have a next O that's hooked up to our next carbon. So that's an ester linkage there, all right? So that is if you want to draw an ester linkage. And this is what and what do you think this is called? This is an example of a triacylglyceride. So this is a tag molecule. So this whole thing here, well, sorry, should I really? Let me be careful. It should be really this, and not the water molecule, but this. This is a tag molecule. All right, where you have it's triacylglycerol. All right. Now, just like with fats and carbohydrates, just like how we could use a condensation reaction to make the triglyceride, all right, to make the tag molecule with all these ester bonds. We could do the reverse, like a condensation reaction will make the triacylglycerides or the tag molecules. It means that we could also reverse the process, all right, if we hydrolyze the tag molecule. Now, in the human body, we would use enzymes, and these enzymes that hydrolyze tag or lipids are called lipases. So basically, it's just a simple reverse reaction taking place here. So when you hydrolyze a tag, you're going to get 3 fatty acids and 1 glycerol molecule. So that's your recap. In the human body, we're using an enzyme called lipases. And basically what a lipase would do is it will hydrolyze the ester linkages, right, to give you 3 fatty acids and 1 glycerol molecule. So it didn't draw them 3 times, so let's put a 3 here. So this means it's 3 of this fatty acids, 3 fatty acids plus 1 glycerol. Alright, now in the lab now, we could also hydrolyze triglycerides. But we could, instead of using an enzyme, we could use a strong base such as sodium hydroxide or potassium hydroxide. And if you do that, you're going to get the sodium or potassium salt or the fatty acid. So you see triglyceride and you see sodium salt or the fatty acid. Normally it's we use stearic acid. Alright, we combine stearic acid, 3 molecules of stearic acid with glycerol. So it means that when you hydrolyze that tag molecule, you would have gotten sodium stearate. And sodium stearate is used to make soap. Alright guys, so we have come to the end of this lecture. So, you know, the normal stories. Check us out on Facebook. Do a search for BiochemJM. We would love, especially international students, we would love to have you on. You know, we always talking about biochemistry and school in general, so we would like to hear what's happening on your side of the world. If you have any kind of images or notes that you think will be of additional help to what you saw here, please send us it. We love to share info back and forth. Alright? And just remember, like with anything you're studying at this level, you should be always reading. You should be always trying to get new textbooks, because the more you look at the same thing at different angles, the better you will understand it. And I hope that when you viewing these lectures, my videos, you have a pen and a paper with you, and you keep making notes along the way. My advice to you all is that you always keep making notes and make summaries and stuff like that. And what you do is that you pause, you rewind, and see if you got everything. Because it's only when you start writing and in your own handwriting, that's when you're going to really understand this material. So keep writing and keep studying. And as usual, if you enjoy this video, show your love, hit the like button on YouTube. And as usual, I give you very hearty thank you for looking at this podcast. Best of luck in your studies, people. See you soon.